All right, hi everybody. So today we're going to start talking about vision. But before we start, I wanted to talk briefly about something else. So on Friday with the climate strike, right, and a lot of people missed class. And before that, I'd actually never even considered the possibility that I could record lectures while I'm giving them. But then I was thinking about it afterwards, and it was pretty easy to do. So I decided that as a trial, I'm going to just try recording all of them and then posting them afterwards. See how it goes. So my intent, though, I just want to get out there that this is not to encourage people to miss class, but just to give you an extra tool for trying to, go back and study or whatever. And I hope also that this is something that allows you maybe to take a breath during class and try to process things more rather than trying to frantically write everything down. We'll see how it goes, and I'll try to check in to see whether it's useful down the road. So this is all pending the technical sort of aspects of it working out, but I think it should be okay. All right, so today we're going to talk about vision. And this is a sense that you've probably heard quite a bit about already. In particular, you've probably heard about the eye, like the structure of the eye, how rods and cones actually detect light changes in terms of rhodopsin and that sort of thing, and how color vision is encoded in the eye. And so we're not going to talk about those things in any depth, but I assume that you've heard of them before. Just to remind you, this is an eye, and light comes in through here, right? And it eventually hits the retina. The retina is what we're going to be interested in today. Okay, what's going on there? And in particular, I want to point out one area of the retina called the fovea, which is sort of like the point of fixation where the light hits your retina from whatever you're fixated on, right? So if I'm looking at the projector, then the light from the projector is hitting my fovea essentially. This is an image of the cell types within the retina, which is the major focus of today, of course. And I want to point out a few things. One is the fact that I'm sure you've heard about before, which is that light actually passes through all of these cells before hitting the rods and cones. So you would think that light would come in here directly and hit the rods and cones, and then it would be processed sort of moving away from that, but light actually passes through all the other cell types, and the rods and cones are actually at the back of the retina, and then the signals from them are sent more forward through bipolar cells, which we'll talk about, and then ultimately to retinal ganglion cells, which are the cell type that transmit all of the visual information to your brain. Okay, there's a lot of different types of retinal ganglion cells. We'll talk about the most famous of them today, but ultimately all visual information goes through retinal ganglion cells to your brain. Okay, there are also horizontal cells that are inhibitory and perform lateral inhibition, which we'll also talk about later. As I mentioned, we are not going to talk about how the rods and cones actually detect the light itself, which I'm sure you've heard of before. I put up this slide only to illustrate a really important fact that you have to get straight in your mind if you're going to understand the rest of what happens today, which is that contrary to what you would intuitively think of happening, darkness actually causes depolarization of these rods and cones, whereas light causes hyperpolarization, right? Normally we think of stimuli as eliciting depolarization, so you would intuitively think that light should depolarize these cells, but it's the opposite, okay? And they are strictly utilizing graded potentials in the rods and cones, and we'll see that later as well. So there are, of course, two types of pigment cells in the retina. These are the rods and the cones. You probably know the cones for their function in color vision. So that is one of the major sort of functional specializations of these two cell types. And rods, as you know, are kind of, not underlying color vision and sort of more involved in, detecting low levels of light and things like that. In terms of, in terms of when you actually utilize your rods and cones, this is an image from the textbook just showing the sort of types of illumination levels that exist in the environment and the types of visual functions that accompany them, right? So if it's very dark out, like you're out in the country or whatever, and there's just like, you can see the stars really well, but otherwise it's really dark, that is called scotopic vision. Then if it's a little brighter, right, so you're under moonlight or you've got some light pollution in the city or whatever, that's more mesotopic vision. And then upwards of here, which includes both indoor lighting and outdoor, like sunlight, you have photopic vision. Okay, so typically the rods are involved at these lower levels of illumination, and then the cones start kicking in at mesotopic and then go through photopic vision. This is, of course, why you can't really see in color when the low levels of illumination are very low. So there is a trade-off there. One other difference in the sort of organization of the rods and the cones within the retina is the connectivity between rods and cones and their downstream bipolar cells. Okay, so this is illustrating that every rod bipolar cell receives input from about 15 to 30 rods, whereas a single individual cone bipolar cell receives input from only a single cone. And this has important functional consequences that I want us to think about for a second. So I have a clicker question here that just asks, what would you expect to be the relative signal to noise and spatial acuity of rod bipolar cells versus cone bipolar cells? Okay, so maybe 10 more seconds. All right. Okay, most people pick C, which suggests that the signal to noise is higher in rod bipolar cells, and the spatial acuity is higher in cone bipolar cells. That is correct. Does somebody want to try to explain why they picked that? Most people picked it, but for those who didn't. Starting with signal to noise, what's the key feature, what's the word to describe the key feature that we see here that we've learned underlies higher signal to noise? Yes, convergence. Perfect. Okay, so we see convergence of all of these rods onto a single bipolar cell, and that is convergence is a mechanism by which you can increase signal to noise. And you should go back and look at the in Friday's lecture to see that if it's not totally clear. And I want to point out something here that I wanted to say on Friday, but I think I forgot, which is that one really common misconception when we're talking about the effect of convergence on signal to noise is that a lot of students will tend to think that it's actually the lateral inhibition that increases signal to noise, but that's not true. It's the convergence, okay? So here we have convergence. It is increasing signal to noise because essentially that downstream neuron is able to pool the inputs from multiple upstream neurons and therefore filter out some of the noise that occurs in individual upstream neurons and only respond when, you know, there's sort of like a consensus response across the upstream neurons. Okay, on the other hand, cone bipolar cells should have higher spatial acuity, in part because, you know, they're receiving information from just like a single little part of the retina, whereas rod bipolar cells would be receiving information from a larger part of the retina. Okay, so this reflects like the size of a receptive field, which again, we learned in the somatosensory system is a direct cause of differences in spatial acuity. Any questions about that? All right. So one of the really interesting differences between rods and cones in the retina as well is their distribution. And so if you look at the density of rods here shown in purple across the retina versus cones shown in green, you can see that in the peripheral parts of your retina, you have a lot of rods and not many cones, okay? Whereas at the fovea, right here in the center, so this is the fovea, you have this spike in the density of cones, and you have a lower level of rods. Okay, so this is really important because this really increases your spatial acuity at the fovea. All right, which means that whatever you're fixated on, whatever your eyes are fixated on, you'll have higher spatial acuity for that area in visual space than you will around the periphery. And this is why, for example, when you're reading, you don't just like hold a book here and like stare forward and then like read right across, right, with your eyes like fixed forward. You could do that if spatial acuity was the same all the way across your visual field, but it's not, right? It's highest where you're fixated, and so that's why you fixate on the individual words as you read across. Okay, if you try to just stare ahead, you can see the words in front of you, but not so much on the edge of the pages. That can be illustrated here as well. So I don't know how well this works at the back of the room, but you can look at the slides later if you want and look at them closer up. The phenomenon that you should see, and I hope most people will see this at least near the front, is that you can see these black dots at certain positions, but when you fixate on them, you know, you can see them really well, but you can't see the other ones so well, right? So they sort of disappear when you're not fixated on them. My own perception is that I generally see the lateral ones okay, but not so much the up and down ones. But anyway, I don't know whether that's true in general. Do people see that? Yeah, it still works? Okay. It's like a long room with a small projector. Anyway, so yeah. So any questions about rods versus cones, spatial acuity, et cetera, et cetera? No. So I mentioned as well that cones express different photopigments, and they underlie color vision because you have different pigments and different types of cones. This is illustrating that fact. And again, we're not going to talk about how that works in this class. Okay, so there's a learning objective to explain the functional differences between rods and cones. For our purposes, you know, we would be more interested in kind of like the signal to noise, density, that sort of thing with respect to spatial acuity. Okay, so now we're going to get into the real focus of today, which is how these circuits in the retina process information as it comes in. Okay, and so just to remind you of the sequence of cells, you see the photoreceptor cells, the rods and the cones out here, and then they signal to bipolar cells, which synapse with retinal ganglion cells, which send the information to the brain. Okay, so we're going to start simple with the types of responses that are typically observed in retinal ganglion cells. And the most famous kind of examples of responses are on cells and off cells, okay? And so let's say you have a retinal ganglion cell, and this is its receptive field, okay? In this case, the receptive field refers to the area in visual space that that particular retinal ganglion cell is responding to, okay? And as you've learned prior, I'm sure, there is the center of the receptive field, and then there's a surround. So let's talk about the center first. And there are two basic types of these retinal ganglion cells. One is called an on-center retinal ganglion cell. And so if you have a situation where, let's say, everything's dark, and then a light appears in the center of that retinal ganglion cell's receptive field, the on-center cell will increase its firing rate, whereas the off-center cell will decrease its firing rate. Okay, so that's illustrated here in the sense that, you know, an on-center cell, you have these little pluses in the middle. That just means that like light in the middle will excite these cells, right? Light in the middle of the receptive field will excite them. Off-center cells are the opposite. Light in the center will inhibit them, and that's why there's minuses there. Off responses are the opposite, right? So if you have, let's say, a illuminated area, and then you just darken the center, then what you see is that the on-center cells stop firing, or they decrease their firing, and the off-center cells increase their firing. So can anyone suggest a reason why you would have off-center cells and on-center cells, instead of just having one cell type that increases its firing when the light gets higher and decreases its firing when the light gets lower? Yeah.
Increased contrast.
Can you explain that?
Like, say that there's like like a blank square on a table or something, and the table's blue color.
Uh-huh.
The borders of like the dark object, they activate the off-center cells more, and then that difference in firing between the on and off-center can like help sharpen the contrast between those two.
Yeah, that's great. And we're going to talk more about like edge detection and contrast a little later, probably today, hopefully, or maybe on Friday. In a more general sense, what he says is totally right. But to sort of step back and even be more general about it, when you're thinking about the efficiency of neural coding, you want to think about like higher efficiency essentially means that there's more information content per spike or per amount of time, right? And so let's say you had only on-center cells like this, right? If we look back here, they're pretty efficient at encoding increases in brightness, right? Because you've got like some spike rate at whatever level whatever level of illumination you're existing at. And then the center of the receptive field gets brighter, they'll increase their spike rate. And increases in spike rate are pretty efficient to detect because there's a lot of information content in a relatively small amount of time, right? So you go from like very few spikes to within, several milliseconds, you're having more spikes, okay? So that's really easy to detect. On the other hand, let's say you have this situation where it gets darker. You go from like one spike every once in a while to like one spike every longer while, and it's not super efficient, right? Like you can't tell very easily or quickly that you've actually had a change because there's much less information content in that change. All right, whereas the off-center cells are now increasing their firing, so you see a big change in their response and to tell you that it's getting darker. Yeah. Yes. Yeah, so the question is, does darkening the center, is it the same thing as illuminating the surround? Essentially, yes, but let's hold that until we talk about the inhibitory surround more. Okay, so there was a learning objective to describe and explain the mechanisms underlying on-center and off-center ganglion cell responses. That's what we're going to talk about now. But just before we go there, any more clarification about the phenomenon of their responses? Yeah.
So on the previous slide, like right after the darkening ends,
oh yeah,
it seems like
there's a rebound. Yeah. So you're talking about here, right? There is a rebound when you go back. That so essentially, like T2 should be equal to T0, but you're noticing that actually there's more spiking here because you're getting brighter, even though the absolute levels are the same at T0 and T2. So this reflects the fact that it's not only encoding the absolute levels, but also the change in the levels, right? So you're going from darker to brighter here, and so it's encoding that change. And there are basically two mechanisms that can underlie this. One is just a physiological, like cell autonomous mechanism within the photoreceptor cells that actually create this rebound just through the signaling pathways that detect light that we're not really talking about today. And then the other one that can contribute to this is lateral inhibition, which we'll talk about later. Yeah.
Does it get perceived as like essentially all of a sudden brightens up?
Well, yes. I mean, in a sense, yes, because you're getting brighter, right? So in a relative sense, it's detected as an increase. But like the absolute level, would you perceive the absolute level at T2 versus T0 to be different? I don't really know the psychophysics on it. Presumably for a small period of time, yes, but I'm not totally aware of the psychophysics. But we'll see an example where the same sort of phenomenon occurs over sort of space rather than time later, where you do perceive the same level of illumination to be different based on like edges and stuff. So yeah, this is just sort of like a temporal example of the same sort of thing. Any other questions? Okay, so these are the circuits in the retina again, and with a little bit more molecular information here. So I want to get some things on the table to start with. One is that there are two types, in addition to the two types of retinal ganglion cells, so we have an on-center cell here depicted as well as an off-center cell. Each of those has a corresponding either on-center or off-center bipolar cell. Okay? On-center and off-center bipolar cells all receive information from the cones. We're just using cones as an example here, right? But the big difference between them is that on-center cells express a receptor called mGluR6. Okay, this is a metabotropic glutamate receptor, whereas the off-center cells express AMPA receptors. So let's think about the off-center cells first, actually, okay? So if the center cone is releasing glutamate, which is the neurotransmitter that they release, it's just like pumping out some level of glutamate, okay? That glutamate is going to depolarize the off-center cell, right? Because it binds to AMPA receptors. We know that AMPA receptors, have an EREV of 0. They will cause depolarization, right? On the other hand, mGluR receptors, this is a metabotropic receptor that actually causes an inhibitory response, okay? So it essentially reverses the polarity of the response in the previous cell. And that's why it's shown here as a minus versus a plus. In the textbook, you'll see them refer to sign-conserving synapses, which are the pluses, which just means that if the presynaptic cell depolarizes, then the postsynaptic cell will depolarize, right? Whereas the negative is called a sign-inverting synapse, which just means the presynaptic cell depolarizes, means that the postsynaptic cell will hyperpolarize, right? Which is essentially just an inhibitory response. Okay, this is through the modulation of cyclic GMP-gated channels. So glutamate binds to mGluR6. That actually decreases cyclic GMP levels in those bipolar cells, and that leads to the closing of these cyclic GMP-gated ion channels. Okay, and the closing of those channels means less inward current. Less inward current means hyperpolarization. All right. So let's look at an example here where the center of the receptive field is becoming brighter. In this case, as we talked about earlier in the lecture, the cone cell will hyperpolarize. Okay, so then the challenge from there is just understanding how that gets translated in the two pathways. Okay, so what we have here is this hyperpolarization means that there will be less glutamate released by that cone cell. All right, because depolarization will release more neurotransmitter, hyperpolarization will release less. If you release less glutamate, that means that there's less glutamate binding to, let's say, the AMPA receptors, which will cause the bipolar cells in the off-center stream to hyperpolarize as well. And you can see right here, right, this is the sign conserving synapse, right? The postsynaptic cell does the same thing as the presynaptic cell. On the other hand, we see here the sign inverting synapse. So here, hyperpolarization of the cone cell means it releases less glutamate again, but this time less glutamate means less inhibition on that off-center bipolar cell, and less inhibition means depolarization of that off-center bipolar cell. Any questions about that so far? Is this basically clear? Because this is the key, right? The difference between on-center and off-center happens at that synapse. Because from there, each on-center or off-center bipolar cell just has a sign conserving synapse with their downstream retinal ganglion cell. In each case, they release glutamate, which binds to AMPA receptors in the retinal ganglion cells. And so depolarization in the off-center bipolar cell, in the on-center bipolar cell, will cause depolarization of the on-center ganglion cell, which will then cause that retinal ganglion cell to start firing action potentials more frequently. Okay, so as you'll see, the cone cells and the bipolar cells are only utilizing graded potentials. The retinal ganglion cells are firing spikes. Okay, so depolarization causes more spikes. On the other hand, the off-center stream here, you see hyperpolarization of the off-center bipolar cell, which leads to hyperpolarization of the off-center ganglion cell, which means fewer spikes. Okay, questions here? This is the kind of thing, and especially when we get to the horizontal cells, where looking at this in the textbook and sort of thinking about it again, rereading it, is going to help if it's not entirely clear now. Sometimes it just takes a little time to sink in. But in any case, this is how these increases in light in the center of the receptive fields cause the increase in the on-center cells and decrease in the off-center cells. Yes?
Mind going over what happens to the bipolar cells when there's less glutamate in releasing it?
Sure. I have a suggestion. Why don't we do the offer responses? And if you still want me to redo it, I'll do it again. But it's basically the same story in reverse, so it might also help. Okay. So let's say now we're darkening the center of the receptive field. Everything happens in exact opposite polarity. Okay? So now the center cone is going to depolarize for reasons that we didn't talk about today, but I told you to accept as fact, right? So depolarization of the center cone cell means that now there's more glutamate released by the center cone cell. Okay? So that more glutamate is going to, through the sign conserving synapse, depolarize the off-center bipolar cell. Right? And so that's what you see here. On the other hand, more glutamate binding to the on-center bipolar cell is going to increase cyclic GMP levels, which is then going to increase the cyclic GMP gated channel conductance, which is going to then depolarize these hyperpolarize, yes. Right. So we have more, okay. So the confusing thing, and this comes up every year, is that the negative, it's hard to remember that like the negative and positive here are the sign of the synapse and not like the change in glutamate release in this particular scenario. But anyway, so in this scenario, right, we have increased glutamate levels, which cause then inhibition of the on-center bipolar cell, right, through mGluR6. Okay, and so then depolarization of the on-center bipolar cell of the off-center bipolar cell will cause spiking in the off-center ganglion cell, whereas hyperpolarization in the on-center bipolar cell will cause less spiking in the on-center ganglion cell. Okay, so since I actually maybe added to the confusion of the mGluR responses, I will go back here and just say it again. Is that still, you still would prefer that? Okay, so, all right. Binding of glutamate to this mGluR6 receptor causes a decrease in cyclic GMP levels. Well, it causes a decrease in cyclic GMP levels, which causes hyperpolarization. That is binding of glutamate, right? So in the case of less glutamate being released, which happens in this scenario, the opposite happens, right? So less glutamate means we have increased cyclic GMP levels, which leads to depolarization, right? Is that okay now? All right. Any other questions about this? Yeah.
Sorry, you said less glutamate depolarization of the on-center cell.
Less glutamate means depolarization of the on-center cell. Yes.
So the next slide, why did the on-center cells hyperpolarize?
Because you have more glutamate, right? So you have the center cone is depolarizing, which means more glutamate, and that causes inhibition of the on-center bipolar cell. Yeah, so the key here is separating, this is what happens here in response to glutamate. So if glutamate goes up, then this is what happens. If glutamate goes down, then the opposite happens, right? All right, other questions? Okay, so this inhibitory surround. If you have a point of light and it starts out in the center here, then an on-center cell would be expected to have an increased firing rate, right? That's what we just learned about. But as the point of light goes outside the center and into the surround, then what you have is inhibition. Okay, which means is reflected by a decrease in the firing rate. Similarly, let's say you have a point of light in the center of an on-center ganglion cell, shown here, that will have a certain firing rate. If you increase the amount of light, the firing rate will go up. However, if that point of light then gets larger and larger and starts also hitting the surround, then spiking goes down. And if it gets really big, then spiking really goes down because of all the inhibition. Okay. So this is how the inhibitory surround works. It's all via these horizontal cells that release GABA. Okay, and so GABA is an inhibitory neurotransmitter, of course, right? And so it will, utilize what we would call here sign inverting synapses. Okay, so the thing you have to keep in your mind here is that each cone cell has a sign conserving synapse with the downstream horizontal cell in that direction, right? But then each horizontal cell signals back to the cone cell and also surrounding cone cells via a sign inverting synapse. All right, so horizontal cells will, hyperpolarize in response to a hyperpolarized cone cell, but then that will lead to the opposite response back on the cone cell. So we'll see how that works here, okay? So let's imagine we go from T0, which is just the low level of illumination, to T1, which is what we've seen already. The center of this receptive field gets brighter. As we learned, this causes hyperpolarization of the center cone cell, and this will lead to depolarization of the center bipolar cell, right? So we should be okay with T1 to T2 here. And the only added thing is that because of their sign conserving synapses with the horizontal cells, you'll see that hyperpolarization of that center cone will also cause hyperpolarization of the horizontal cell. Okay, so here's the key thing. When you get light in the surround, that's going to cause hyperpolarization of all those surround cones, which means that you also get hyperpolarization of all the surround horizontal cells. And so that's what we see after T2 here, where these horizontal cells get even more hyperpolarized. Okay, so here we can write that the surround cones hyperpolarize. That's not spelled right. Hyperpolarize. And then that leads to hyperpolarization of those horizontal cells. Okay? So now hyperpolarization of the surround or the horizontal cells means that they will then release less GABA. Okay? And if you release less GABA, that means that there's less inhibition back onto the cone cell. And we see that then the cone cell will depolarize. All right? So in the absence of these horizontal cells, you would expect like the cone cell to stay hyperpolarized because there's still light in the center, right? But then these surround responses, which cause the hyperpolarization of the horizontal cells, releases the inhibition on those cone cells, which then causes depolarization. All right? That doesn't mean that they're depolarized, beyond where they started. It's just depolarized relative to what they were when you had the light in the center. All right? And so that's why you see this happening in T2. And so what this does is it just gets then read out in the bipolar cells, right? Which in this case, they're showing an on-center bipolar cell. So this essentially inverts all the responses from the cone cell. So now we have the depolarization during T1 and then more hyperpolarization during T2. And then the corresponding on-center retinal ganglion cell, you see, will fire a lot during T1, but then fire less during T2. So you can imagine now that if you were to darken the surround everything happens in the opposite direction, right? If you darken the surround, that means that the surround cones will depolarize, which means that the horizontal cells depolarize, which means that there's more GABA, and more GABA feeding back onto that center cone is going to then hyperpolarize the center cone. So as I mentioned, this is really the kind of thing that you have to sit with for a few minutes and just work out the signs of everything. So don't feel like worried if it's not all crystal clear right now. But having said that, do feel worried if you can't figure it out after looking at it for a while, and definitely come ask me about it. But for now, are there any other questions that I can clarify before we move on? Okay. So, you will definitely see a question about this stuff on midterm number 2. So yeah, be ready. I'm sure if you look at the practice exams, there's a question about this kind of thing on all of them. Okay. So edge detection was brought up earlier today when talking about the on-center, off-center responses. And so we're going to just spend a couple of minutes talking about why the center surround model actually enhances edge detection. Okay, so imagine that you have a bunch of on-center retinal ganglion cells that have receptive fields sort of across visual space for a particular area, right? And you have in that area an edge between dark and light. Our visual system is particularly good at picking out edges, okay? And part of the reason is that there is the strongest contrast between retinal ganglion cell firing at this edge. So that can be illustrated here. If we look at the response rate of the on-center retinal ganglion cells, and let's arbitrarily just say, A is here, we'd see that B, which has part of its surround in the light, is going to have a smaller response. C, which now has, part of its center in the light, but also more of its surround in the light, maybe comes up a little bit again. Whereas D, which has its center in the light, and then some but not all of its surround in the light, is going to have the highest firing rate. Whereas then E, where you have the center totally in the light, but now the surround also totally in the light, it's going to go back down. So you can see that the difference between A firing and E firing is actually pretty small, right? Like the difference between absolute levels of firing, if you're looking at a dark area versus a light area, is not that big. However, the difference between B firing and D firing is very large. Okay, and these are the retinal ganglion cells that sit at that edge between the light and the dark. That's where the highest contrast is between the firing of these neurons, and that's why that edge is like the most easy to detect. We can see this phenomenon of surround inhibition and the effect on edge detection in a visual effect called Mach bands. So if you look at this image here, these are just circles of increasing luminance going from the center out, okay? So if you looked at the luminance, they would look just like this. They are steps, okay? So within every band, the brightness is the same across the band. I know this because I made this diagram, so I can guarantee that within every band, the luminance is the same. But if you look at this, right, every band looks brighter on the inside and darker on the outside. Okay, that's a phenomenon called Mach bands. And the reason is this. If you have a retinal ganglion cell that has its center here and its surround also within that band. Oh, the contrast on this is super weird. You can't even see the... Okay, there's another band here, in case you can't see it. It's just a little bit lighter. Okay, so that'll have, you know, a certain level of firing. But if you have a cell that's here, has the same amount of light in the center, but it has less inhibition because there's less light in the surround, the firing rate of this one will be higher. Okay? Whereas here, we have the opposite. We have the same amount of light in the center again, but we have more inhibition in the surround, and so the firing rate is going to be lower. Okay, so these are 3 points in space where the center receives the same amount of light, but there's different amounts of inhibition from the surround, and that's why the retinal ganglion cells in this area fire more, and therefore you perceive that area to be brighter, whereas the retinal ganglion cells in this area fire less, and you perceive that area to be darker. Okay, so we are out of time, so I'm going to stop there and think about this. If you have questions about it, ask me at the beginning of Friday.